Okay, now thank you very much, Marcus, for inviting me for this. Now, I apologize that I'm not there. As Marcus already mentioned, I got tested positive on COVID last weekend, and I'm doing okay, but still not perfect. I really miss this meeting that you are at, seeing everyone again and physical contact with everyone again. Is very nice, I think. So, but still, I'm very pleased that I can join this workshop. And yeah, I apologize that you cannot see me in real, but I still want to talk a little bit about work that we have been doing together with MS Vision. We work already for a very long time with MS Vision. Where we'll talk about today is more recent work that we did together with MS Vision, but also with Thermo Fisher, also with e-MSion, the company that makes these ECD cells. And so I'm going to talk about how we use ECD and how we implement it on different mass spectrometers and how it can be beneficial to really look at top-down sequencing of antibodies. And so before I continue, I really want to thank the two postdocs who did most of this work. One of them was Kyle Ford, who started with this already 4 or 5 years ago. And I must say the first ECD cells we got from e-MSion, they did not work very well. The filaments burned all the time. So I really want to thank him for putting in all the efforts to get this better. And then more recently, and I should say Kyle Ford is now working for Thermo Fisher. And more recent years, Jean-François Grice did all the work that I showed today, also a postdoc. And he also left the group and he works now for Bruker. So if you start to work with ECD in my lab, it seems that you end up at some of these big companies. Why are we interested in this ECD? That is because we want to study antibodies. And yeah, if you don't know that much about antibodies, there is a zoo of different antibodies in our blood or in our bodies. Here you see just a picture of them. We all know now what they do. They recognize pathogens and help our immune system to get rid of these pathogens or infected cells. But if you look more into detail, there's a whole different array of antibodies. And here you see some of them. The most prevalent ones in our serum are the IgGs, and then especially IgG1 and IgG2. But also IgA occurs a lot in serum, but also in human milk. IgM occurs a lot in serum, and you see some others that are maybe less abundant. Now, they are all different, but they also share quite a bit of structural features. And especially these arms that you see over here, you see them coming back all the time. They are called the Fab arms, and they are actually the antigen recognizing active sites of the antibodies. And yeah, that is where we are most interested in. This part of the antibodies is called the constant region, the FC region. And as the name says, that is way more constant. This part is not constant at all. So if you zoom a little bit more into that, here you see the structure of an IgG antibody. Here's the FC region that also carries an end glycosylation. And this is a cartoon of the Fab region. And you can actually split this Fab region from the constant region by using an enzyme called IDS. And then you're left over with the part that carries all the antigen binding sites and is this region that you see over here. Now, this cartoon color codes several different things. In colors here are the different CDR regions, and these CDR regions are really the regions that are recognizing the antigen, but that are also hypervariable. And in blue, you see, for instance, disulfide bridges. And in the hinge region of the Fabs, there are also disulfide bridges, and they can bridge these antibodies parts together. Now, you see, for instance, already in IgG1, there is a disulfide bridge here. But in IgG2 and IgG4, this disulfide bridge is not formed. Instead, there is a disulfide bridge formed from here to there. So in that sense, it's already a different molecule. Now, I'm really interested in antibodies and especially about serum antibodies in our body because the question is, how many are there? Now, these antibodies, they are made by distinctive B cells in our body. And it's estimated, and you can look at these numbers over here, that there are maybe about 10 to the 9th, so that's 1,000 million different B cells in our body. And each of these B cells makes another antibody. And how can that be done? Now, maybe you know that from immunology textbooks. Actually, especially these CDR regions that you see over here, they are made by recombinations of V, D, and J sections of genes. And by a plethora of different combinations, you can end up with a combinatorial combination of different gene sequences. Now, this is one way to look at it. Another way to look at it is if you look at the sequence of different antibodies, like in the IMGT database, you see that at some sites, they are very conserved. And here you see, for instance, a totally conserved region, while other sites are really hotspots for mutations. And these hotspots you see in the CDR1 region, in the CDR2 region, and the CDR3 region is most notable known for all these different mutations. So the point I want to make, there are parts of the antibody that are hypervariable in sequence, and there are parts in the antibody that are more constant. Now, if you want to do antibody sequencing, why should you sequence the constant part of an antibody if you know already what the sequence is? Now, you're mostly interested in these hypervariable regions. They define the uniqueness of that specific antibody. They define the antigen specificity. And of course, that is the information that you want to know. So we use for this, to sequence these antibodies, we use different methods. And of course, you're all familiar with CID and HCD, generating B and Y ions. But we use now primarily ECD, and this stands for electron capture induced dissociation. And the interesting thing is that this breaks the peptide backbone in a different part, and it breaks them really into C and Z ions. Now, what we do is we take this little cell from e-MSion. It's really small, and we can put it, in this case, in an Orbitrap mass spectrometer. And you see we put it here in between this lens and here the cell to the Orbitrap, and it's just placed here in between. So we can do mass selection. Then we do ECD here in the cell, and then the ions can go into the HCD cell, they can go into the Orbitrap. Then every ion that comes in here can be selected and can be subjected to dissociation by this ECD fragmentation. Now, what is a nice thing about this ECD is that it breaks these C and Z ions, but in particular for antibody molecules or for Fab fragments of these antibody molecules, it breaks them specifically in between this little region and that little region. And you will see later on that we get specific fragment ions of this particular part of the molecule. You might say, why doesn't it fragment at other places? It does. But if it fragments at other places, and we do this under native conditions, there are disulfide bridges that keep all the fragments actually together. So only these N-terminal fragments, this one on this side, but also this one on that side, are the only ones that can really fall off after ECD fragmentation, and that are the ones that we detect. And I want to show you that this is a very powerful method to get sequence information, especially about this region. And fortunately for us, this region actually covers part of this CDR region that we are interested in. Now, how does an ECD spectrum look like? Now, I first should tell you, here's the precursor mass. And mostly what happens under ECD conditions is that the precursor ion captures an electron, another electron, another electron, another electron, another electron, another electron. So we see here electron capture without fragmentation. However, what we also see is fragment ions that are here at the low M/Z region. And because we have relatively high resolution, we can blow up that part. We can actually deconvolute the mass spectrum, and we see a beautiful range of fragment ions over here. These are all the C ions. The concomitant Z ions are actually the remainder of the molecule. So that's a very big mass. We can detect that as well, and that are the peaks that you see over here. And here's the mirror plot of them. They are much less well resolved, so we actually only look at the C ions in the low M/Z region. Now, this is an ECD spectrum of a monoclonal antibody, Trastuzimab. And by using this specific enzyme, we cleaved off the FC region to generate this Fab2 molecule. But, and that I show over here, we can equally well do this on the intact antibody on the Fab2 that I just showed you, or on the Fab molecule. And this we can make by reducing the Fab2. Now, if we do this, we see here the fragmentation patterns. And because we know the sequence, we can actually nicely annotate them. And the beautiful thing is what we observed is that for both the heavy chain and the light chain, we get a beautiful sequence tag of the whole sequence of this antibody in that particular region. Now, this region for the light chain covers the whole CDR3 region, where we are most interested in, and also some of the framework regions. For the heavy chain, we also cover the whole CDR region and also some of the framework region. So we don't get 100% sequence coverage. That's maybe also not what we want to get, but we get very important information about the variable sequence of the antibody, and both of the heavy chain and the light chain, because we have them still paired. Furthermore, you see that we can almost annotate all the fragment ions in the ECD spectra. They are extremely clean. And actually, we do this with software, but the spectra are so clean that you can almost annotate them manually. So this is for IgG. What we next did is where are these fragment ions? Now, they are really in this CDR3 region on the structures of the antibodies. But we also thought, okay, if this works for IgG1, can we do the same for IgG2, IgG3, and IgG4? And I remind you again, these molecules look the same, but for instance, their disulfide patterns are completely different. So we looked at those again, and I go through this quickly. But this is the IgG1 Fab2. This is the IgG2 Fab2, the IgG3 Fab2, and the IgG4 Fab2. This is all each the same antibody. And we see exactly the same sequence tag. So we just can show you now that we can sequence any antibody of each isotype with this ECD method. Now here you see the differences in the spectra, and this was all the same sequence, but different antibody formats. And you can see that the fragmentation spectra that we get are extremely clean, extremely reproducible, and extremely annotatable. Next, and I find it actually a bit more exciting even, we said, can we also do IgA? And so we made an IgA having the same sequence as the IgG in its variable regions. But if you look at IgA, there is also a huge difference between an IgG and an IgA molecule. And that is not only in the disulfide bonds, but also IgA is heavily N and O glycosylated. So if you took a mass spectrum of an IgG, as many have done by you, or native mass spectrum of an antibody, it looks quite familiar. If you take a similar native mass spectrum of an IgA, it looks like this. It looks horrible. You cannot resolve the charge states anymore, so you actually don't know where you're looking at. Still, what we did is that we took a precursor here and did ECD, and we took a slice of this blob and also did ECD on those, knowing that this part of the molecule was the same as in here, at least the sequence is the same. Now, nicely, what we get, if we look at the ECD spectra, we see here the ECD spectrum of the IgG, here the ECD spectrum of the IgA. It looks horrible. But if we look at the low MOZ region, as you see over here, we see actually two spectra that almost look identical. So also of this precursor that is not resolvable in the MS1, we get beautiful sequence information in MS2 by doing this ECD under native conditions, generating only the C ions. Here you see the mirror plot again of the IgA and the IgG, and you really see that the spectra are identical. So we can now also sequence IgAs, and this is something that we really wanted to do. We can do this in multiple ways and also for different IgAs. Now, finally, and this is the most recent work, we said, can we also move a little bit further? And can we also do this for IgMs? And can we do this for a whole new antibody format that we are interested in, the non-covalent IgG hexamer? Now here again, your MS1 spectra become totally different. Here you see the native MS spectrum of an IgM, a recombinant IgM. This is a molecule of about 1 million Dalton. And this is the hexamer of an IgG1, also with a mass of about 900,000 Dalton. Again, we select a precursor, and we do MS2 with ECD on these. And again, first the ECD spectra, and here they look pretty horrible because you get this electron capture without dissociation ranging up to a very high M over Z. So I think somewhere here's the precursor and here's all the electron capture. But still, if we only zoom in at the low molecular weight fragment ions, we can really get spectra again that are very nice to interpret, that give us the complete sequence coverage of the CDR3, the framework regions, even while we are doing now top-down ECD on molecules of about 1 million Dalton. So this is what I want to show you. This ECD cell works very nicely for us. We have implemented it on an UHMR, but we also have implemented it on a Synapse instrument. Also there it works very nicely. If you want to read more about this, then these are some references that I can refer you to. And with that, I'd like to thank you for your attention, and I'm very open to answer any questions. Thank you very much.
Yeah, thanks a lot, Albert, for this very nice presentation, which is quite close to my heart because I did my PhD on a similar topic as well. Are there any questions? Yes, please.
Yes, Albert, thanks for the nice presentation. I have a question about the de novo spectra. I mean, for those parts we are talking about, it's very difficult to actually find the sequence if you don't know that. How difficult it is to actually annotate the ECD spectra to unknown sequence of that part?
Yeah, that's thank you very much for your question. I think that's a very good question, and I can answer it in two ways. It's still quite difficult. I always say doing de novo sequencing on antibodies is very easy if you know the sequence. It's a bit more difficult when you don't know the sequence. Saying that, what helps us here and why I'm such in favor of this method is that we get very clean spectra. All the ions that we see here, they are C ions, except a little bit A ions as well that you see here in green actually. Then the next difficult question is, how do we know which ions come from the light chain and which ions come from the heavy chain? Now there, I think we have found a way now here it's done in coloring them purple and red. But now we have a method that we can do this automatically on de novo sequencing. And then I don't think we get a full sequence, but you can clearly see from peak differences here. You can annotate which sequence tags. So you get very easily sequence tags of like 4, 5, 6, 7 amino acids. And that doesn't give you the full sequence, but it allows you to, to filter out different possibilities that you have of the sequence. And in my lab, so this is not the solution, but in my lab, we combine this now with bottom-up analysis of the same antibodies for de novo sequencing. And then these sequence tags really help you to filter out potential solutions that you also get off the bottom-up data. So it's not perfect yet, but I think this is really, if we can get this a little bit better, is really helping us to do de novo sequencing on plasma antibodies. And this is really something that we want to do.
Thanks from me as well for the talk. You had the great luck that in your antibodies you basically could look only at this one dissociating fragment via ECD. Do you think there's a way to implement a similar measure for other protein complexes, for example, by artificially linking them and then looking at parts of the protein complex?
I think that's a great suggestion. Indeed, our luck is here. That we do this under native conditions, so the disulfide bridges are still there. So I think for other proteins, and many plasma proteins have disulfide bridges, you might be similarly, as you call it, lucky. I think, so I think this can be true for other proteins as well. Of course, while we want to focus on antibodies because there are so many different sequences there. The sequencing of antibodies, I think, is a whole world by itself and requires new fragmentation methods and new sequencing methods. And yeah, I think this method will not solve everything, but will really help there as well. But to answer your question, yes, I think you could also induce specific artificial disulfide links or whatever, and they can also induce an only fragmentation that you want to see. So I think it's a great suggestion.
Yeah, I also have a question which is related to that. I was a little bit surprised to see that all the disulfide bridges remain intact during the ECD fragmentation, because usually I would expect that there happens some reduction of the disulfide bridges under the ECD conditions, and the intact disulfide bridges is probably one of the reasons why you get the clear C series on the spectra, because the Z series just shift out of the mass range where you're looking at. Is there an explanation why the disulfide bridges all remain intact during these analyses?
Yeah, that is a very good question. I think this is still a little bit of a debate, even internally in our lab. You know, here the observation is that we only see fragments that are not initially kept together by disulfides. And we don't see reduction of disulfides when we do ECD on native proteins. I know, and also from our lab, there's different work where people do ECD on disulfide peptides, and disulfide bonded peptides. I also want to say what we try to do here, we really try to avoid to also get collisional activation. So we really try to do ECD under very mild conditions, not accelerate the ions at all. And I'm not 100% sure that maybe some of the disulfide breakages that you see can be a sum of electron capture and collision-induced dissociation. So I don't have a perfect answer to your question, but from all the data that I showed you, the disulfide bonds that are present in antibodies really, you know, block the cleavages of other fragment ions, or we don't see them in the fragmentation spectra. So it's our luck here, and I don't want to change that luck, because this sequence information is very important for us.
Okay, thank you very much. I see no further questions in the room here, so I would like to thank you for the presentation. I would also like to thank both other presenters for their presentation, you for your attention, and I would also like to use that opportunity in conclusion to draw your attention to the upcoming IMSC conference in Maastricht. As a Dutch company, we're happy that this conference will take place in the Netherlands, and we would also like to heartily invite you to participate in that conference taking place in late August, early September. Thank you very much for your attention, and I hope you did enjoy the workshop. Thanks.
Thank you very much.